Okay, hello everyone. Thank you so much for joining us and welcome to the fourth iteration, which is the April edition of the GSU Malaria Journal Club, which we run on a monthly basis. Today's webinar is a parasite-focused one, which is going to be referencing Angelina's recent preprint titled, as you can see on the screen, Genetic Diversity of Plasmodium falciparum reticulocyte binding protein homolog 5, which is a potential malaria vaccine candidate, baseline data from areas of varying malaria endemicity in mainland Tanzania. My name is Babalo Chauke, and I'm a training coordinator at the Genomic Surveillance Unit, working with an awesome team of people to organize these monthly events, some of whom are in the room currently. So thank you so much for joining us. Just a few ground rules. The Journal Club is being recorded and will be available on our YouTube channel later today, the Malaria Gen YouTube channel. So please do check it out and you can share with your friends and colleagues as well. Kindly stay on mute so that we don't disturb a speaker. We will have a Q&A session at the end where you can either raise your hand and we'll let you unmute and speak, or you can type your question in the Q&A and chat box and I'll read them out for Angelina as well. Without further ado, I'd like to introduce Angelina Kisambale, who gladly agreed to give this April Journal Club. So quite excited and looking forward to her presentation. The title of the talk is similar to the paper, as you can see on the screen currently. I'm quickly going to read a short bio to let you know who Angelina is and what she's done. And she's done a lot in her career as well as a young ECR, early career researcher in the malaria space. Angelina holds a bachelor's degree in biotechnology and laboratory sciences from Sokoine University of Agriculture in Morogoro, Tanzania, where she graduated in 2021. During her undergraduate studies, she conducted a project on home management of malaria in Morogoro municipality, Tanzania, and generated insight on household prevention and control practices for malaria. In May 2022, Angelina joined the National Institute of Medical Research as a research intern and conducted a 12-month training within the Project of Molecular Surveillance of Malaria in mainland Tanzania, MSMT for short, under the supervision of Prof. Des Ishongoma, who's also in the room currently. From May 2023, she has been working with MSMT project as a research assistant, and meanwhile, she has secured a scholarship to start an MSc training under the support of the second phase of the MSMT project. She's expected to start a training at the University of Basel under the supervision of both Professor Ishongoma as well as Dr. Christian Nsabanzana. Angelina's research interest and recent contribution are in malaria parasite surveillance by mapping parasite species distribution, investigating antimalarial drug-resistant patterns, and studying genetic diversity of malaria vaccine candidate genes. Through her work, she expects to provide crucial insights on the biology of malaria parasites, as well as the effectiveness of current control strategies. Angelina is passionate about genomics as well as life sciences and is dedicated to impactful global health research. She envisions a future where molecular surveillance is integral to health research, strengthening disease control, and supporting the ongoing elimination as well as eradication strategies. Angelina, thank you so much for joining us. You may begin your presentation.
Okay, hi everyone. Thank you, Mr. Pabaro, for a kind of introduction and a short summary of my biography. As I have been introduced by Pabaro, I am Angelina Julius Kisambale. I am working as a research assistant from the National Institute of Medical Research under the project of molecular surveillance in Mainland Tanzania, but also I am under the Pfrh5 Institute, which is funding the second phase of the molecular surveillance of malaria Tanzania project. So I'm here to present the findings of our recent study that was published in Malaria Journal entitled The Genetic Diversity of Plasmodium falciparum reticulocyte binding protein homolog 5, which has been studied previously from different studies and proven to be a potential malaria vaccine candidate. And for our study, the data were drawn from areas with different malaria endemicities from mainland Tanzania. So, starting with the global malaria burden, as it is all known that malaria is a major health public health concern global, with the WHO reports of 2024 highlighting that there were an estimate of 253 million cases with an incidence rate of 60.4 per 1,000 individuals were at risk of being infected with malaria. While, as highlighted on the panel of this map, the red color is the WHO African region, as this region constitutes to the most of the malaria cases that are being reported by the WHO, whereby in 2024, about 94% of the global malaria cases and 95% of the global malaria deaths were originated from the WHO African region. So we can see that this region experiences the highest burden of malaria cases globally compared to other countries. So highlighting on the burden of malaria in mainland Tanzania, based on the recent reports, Tanzania was among the countries that had high endemicity of malaria, whereby about 3.3% of global malaria cases and 4.3% of global malaria deaths were reported being from Tanzania. And as highlighted in this map, 93% of the population in mainland Tanzania resides where malaria is actively transmitted. As you can see on this map, that most of the regions are highlighted red, as indicating that they are originating from areas with high malaria transmission intensities. Though some other regions at the mid-corridor of the country experiences moderate to low malaria transmission compared to other regions. And the possible regions may be due to the environmental conditions within these areas, that they are poor and some are at very high altitude, like Arusha and Kilimanjaro, that they do not provide favorable conditions for the malaria vectors to interbreed and get the parasites to human food. So this is the overview of malaria burden in Tanzania. So since in the last many years, different countries have been put in place several interventions to fight against the malaria. And though despite the interventions, still there are other threats that are hindering malaria eradication. The key interventions that are in place up to date include the vector control interventions such as the insecticide-treated nets, the indoor residual spraying, the larval source management, but also other intervention at the case management, which improves early diagnosis using the rapid diagnostic tests, also using the microscope, but also effective treatments that is done treating the malaria parasites, specifically currently using the artemisinin-based combination therapies, but also injection using the artesunates. Other interventions under case management include chemoprevention, whereby malaria is being targeted during the peak of malaria transmission, so that to block the transmission of malaria, especially in regions that experience seasonal malaria transmission. Other interventions are the surveillance, monitoring, and evaluation, which is done to to overlook, which is done to look for the evaluations that are in place and to target the control efforts that are being put in place. But also the major advancement that has been done so far, it is the vaccination, whereby so far we do have 2 approved malaria vaccines, the RSS and the R21 vaccine that have been recently approved by the WHO. Though despite these interventions that are in place, still there are threats that are hindering the elimination efforts, and these threats include the insecticide resistance, the drug resistance, where the parasites are undergoing resistance against the used antimalarial drugs, but also diagnostic resistance, where some cases are being missed by the rapid diagnostic tests, especially when the parasites have undergone deletion on the histone-rich protein 2 for the rapid diagnostic that depends on detection of the HRP2 or HRP3 genes. The other threat is the presence of invasive vector species. For instance, the anaphylaxis pesticide that has widely been reported in some countries, but also in African countries. So the presence of invasive species also hinders the intervention efforts that are in place. The other threat is the climate change and environment factors. The change in environment factors may shift the transmission dynamics in malaria. That's why making the efforts to eliminate malaria not being reached as expected. So despite these interventions, there are some key milestones that are being put in place since in 1967 that were aimed to develop malaria vaccines and to incorporate these vaccines alongside other interventions. So since 1960s, the scientists have been trying to develop malaria vaccines targeting different stages of the malaria parasites. And so far, up to date, in 2021, the WHO recommended the widespread use of the RTS vaccine after a very long phase of trials in different countries, in different settings with different adjuvants. But finally, in 2021, this was highly recommended to be used by the WHO as a vaccine, especially in children from endemic regions. But also in 2023, another vaccine, the R21 matrix vaccine, which targets the same stage as the RTS, was also approved by the WHO in 2023 to be used also among children residing in high transmission settings. And all these stages target the Plasmodium, Pfrh5 circumsporozoite surface protein, which is the pre-lymphocytic stage of the malaria parasites. But so surprising, in 2024, a study was also conducted in Burkina Faso, where they assessed the safety and efficacy of the Pfrh5-based malaria vaccine among children. And this was a randomized controlled trial among children that reside in Burkina Faso. And the findings revealed that the Pfrh5 had a promising efficacy to be incorporated in next generation malaria vaccine platform. So after highlighting the key milestones that have been achieved so far, still we still need to develop other malaria vaccines. Despite that, we so far have the 2 vaccines that are already in use. But as you can see that the efficacy of these 2 vaccines is not 100%. As you can, studies have revealed the RTS vaccine to have an efficacy of around 40 to 50, but the R21 matrix having an efficacy of up to 75. But the efficacy of all these two vaccines are suspected to wane out with time. So with time, the efficacy of these two vaccines might decline. So we still need to search and develop other malaria vaccines. And the key things that emphasize to develop other malaria vaccines, firstly, to target the highly conserved genes, as when the highly conserved gene is targeted, this can help to advance protection and even enhance the malaria elimination efforts. We also need to develop other vaccines so that to develop a broader spectrum vaccine that will be targeting different strains. As you can see, based on the previous experience, the current vaccines have very low efficacy, have only efficacy that are specific to the Pfrh5 strains. But when it comes to other strains, the efficacy of this vaccine is very low. So we need to develop other vaccines that are able to target different parasite strains. But also we need to develop vaccines that can cut across different stages of the malaria parasite, as the parasite itself is very complex, having different stages in the human host. So when we develop a vaccine that is able to cut across different stages of the malaria vaccine, this can enhance protection and even enhance the malaria elimination efforts. And so the Plasmodium falciparum, the triple-site binding protein Pfrh5, has been studied from previous studies, and it plays a crucial role in the invasion of the erythrocytes. As you can see that previous studies assessed its structure and its crucial role in parasite survival, and observed that this gene is highly crucial in invasion of the melanocyte into the human red blood cells. And the parasites that lack this gene are not able to invade the human erythrocyte. So from its crucial role as a bridge of the melanocyte invasion to the human erythrocytes, as you can see that this gene, it functions alongside other proteins, including the Rh5 protein and the cysteine-rich protective antigen. So after forming a tight junction, they also go to bind to the human receptor of the erythrocyte called the base gene. So the Rh5, it plays a very crucial role in invasion of the parasite into the human erythrocytes, and this can be read from previous studies that assess this structure and its crucial role in parasite survival. So stemming from its crucial role, other previous studies have also assessed its potentiality as a malaria vaccine candidate. Pfrh5, a study by Douglas et al. In 2011, the findings revealed that the antibodies that were induced by the Pfrh5 vaccination inhibited all the Plasmodium falciparum strains in all the isolates that were tested. So you can see that the Pfrh5 is a crucial target for vaccination, even that it is conserved even alongside different Plasmodium falciparum strains. But also, a study by Natam et al. In 2024 that assessed the safety and efficacy of the blood stage vaccine based on the Rh5 among children in Burkina Faso revealed that the vaccine was safe and had high immunogenicity, and their results overall showed a promising efficacy against the clinical malaria. Also, the Pfrh5 has low levels of polymorphism when compared to other blood stage malaria vaccine candidates, and this was revealed by the start this was revealed by a study that was done by Brigade in 2021, where they revealed that there were an excess of rare variants within the Pfrh5 gene. So stemming from these findings, all these findings substantiate that the Pfrh5 is a potential malaria vaccine candidate and outperforms all other vaccine candidates in the blood stage malaria vaccine platform. For our studies on the Pfrh5 gene, we firstly aimed to evaluate the genetic diversity within the gene using samples that were isolated from parasites collected from different geographic settings, but also aimed to generate baseline data on the levels of polymorphism within the Pfrh5 gene. And for our study, the data were obtained from a database on the malaria genetic epidemiology network, which is a free accessible database with multiple of sequence data from different countries, including Tanzania. So for our study, we used the full genome sequence data from Tanzania, and the data were drawn from sites including Mleba in Kagera, Kigoma Ujiji in Kigoma, Muheza in Tanga region, Morogoro Municipal in Morogoro region, and Nachingwea in Lindi. And we retrieved a total of 697 sequences, but after doing the filtering criteria, the samples that remained and passed the quality control were only 589, and most of the samples were originating from Muheza. So for our study, it traced a total of 589 sample sequences that had good quality based on our filtering criteria. So firstly, the gene was extracted from chromosome 4, as it is located on the chromosome 4 of the parasites. So after having the sample sequences on chromosome 4, we did a quality check based on the VCF file, and after doing a quality check, we remained with 589 sample sequences, as I said, and we also called the variants by mapping this sequence data to the reference sequence, which was the Pfrh5, and also variants were filtered using the variant quality score log odds about or equal to 1, and finally we generated the FASTA files sequences, and these FASTA files were further used for analysis of the genetic matrix, and these matrix included the identification of single nucleotide polymorphisms and SNP density, the haplotype and haplotype diversity, the haplotype network, nucleotide diversity, but also we looked for the population structure and differentiation by assessing the right succession index and the principal component analysis. We also estimated the evidence of signatures of selection and recombination by analyzing touch mass and linkage disequilibrium and associated statistical tests, including the FU and RIS test statistics and the FU and RIS F test statistics. So our findings revealed that in Muheza and in Mleba, most of the samples had monoclonal infection, with the FWS being greater or equal to 0.95. Having high monoclonal infections in these regions suggests that there is high parasite inbreeding. But for the sites from Kigoma, Ujiji, Morogoro, and Nachingwea, most of the samples, that is over 50%, had polyclonal infections, indicating that there were frequent infections with parasites of multiple clones. So we're residing in these areas. The assessments of nucleotide diversity and haplotype diversity revealed that there were low nucleotide and haplotype diversity, and the nucleotide diversity was low as 0.0053 in Morogoro and Muheza, and as high as 0.0068 in Nachingwea. And the haplotype diversity was also low as 0.44 in Kigoma and 0.67 in Nachingwea. But the mean nucleotide diversity in this gene was low compared to the nucleotide diversity of the Plasmodium falciparum circumsporozoite surface protein gene. And the results on the nucleotide diversity of the Pfrh5 gene were reported by Limo et al. In 2024, who also used similar study settings and similar data as that which were used to assess the diversity of the Pfrh5 for our study. So, revealing that there were low nucleotide diversity and haplotype diversity, this indicates that the parasites restrict mutations in the Pfrh5 gene so that it is still able to invade the erythrocytes, as perhaps when the parasites allows mutation in this gene, stemming from its crucial role in parasite invasion, the parasite will not be able to invade the erythrocyte. That's why it tries to restrict further mutations in this gene. That's why we see the low nucleotide and haplotype diversity within the gene. When we observed for the haplotype network, we aligned the haplotypes with the reference strain, which was the Pfrh5, and alignment of haplotypes to the reference strain, the 3D7, revealed that this reference strain was found along the haplotype 1, and this haplotype 1 was found in 3 sites, was found in Muheza, in Kigoma Ujiji, and in Mleba. But interesting, 2 haplotypes, haplotype 2 and haplotype 5, occurred in majority of the sample sequences and were shared among all the study sites. As you can see that they accounted for 81.8% of all the monoclonal sample sequences that we are using to assess the haplotype networking within this gene. So despite that some other haplotypes occurred independently in each site, they did not affect much the polymorphism in this gene. So from the observation that some parasites were shared, some being among from all sites, some being among 2, 3 sites, this indicates that there is a circulation of alleles, and the circulation of alleles can be influenced by the presence of gene flow within the parasite populations or sharing of common ancestry among the parasites from different geographical settings. We also assessed the distribution of haplotypes based on individuals with different age groups, and we had a total of 20 haplotypes, and among these 20 haplotypes, 13 haplotypes were found unique to specific age groups, with most of the haplotypes, that 11 haplotypes, were found in children under 5 years, while adults and children 5 to less than 15 years had 1 haplotype each. But it's also interesting, 7 haplotypes were shared between 2 or across all age groups. As you can see here on this diagram, 3 haplotypes were shared across all age groups, while the other 3 haplotypes were shared between under 5 and school children aged 5 to less than 15 years, while 1 haplotype was shared among school children and adults aged above or equal to 15 years, and while there were completely 0% for specific sharing of the haplotype between the 2 age groups, that is between under 5 and above or equal to 15 individuals. So the sharing of haplotypes among individuals with different age groups also still reveals the circulation of haplotypes or the circulation of alleles across individuals of different age groups. We also looked for the population structure within the gene to see how the gene has distinct features from one site to another by plotting the principal component analysis and the light fixation index. The results from the principal component analysis showed that there were no population structure, as you can see on this left panel, that there is some sorts of homogeneous distribution with some outliers, but the diagram revealed that there were no distinctive structures among different sites. But also the rise fixation index that was compared between one site and all sites revealed that there were very low values of the rise fixation index that was close to 0, indicating that the parasites have similar genetic traits. So having a lack of population structure and low values of light fixation index reveal that there is a frequent interbreeding among parasite populations that highly facilitates gene flow within the parasites, thus leading to a homogeneous population structure. When you analyze for the single nucleotide polymorphism SNPs, the SNPs first we observed cases of SNPs across the entire gene, but most of the SNPs were found at nucleotide-based positions around 1,500 to 2,000. Despite that, this region had high density of SNPs, still the gene had low levels of polymorphism when compared to other blood stage vaccine candidates. So it's more likely that the parasite is trying to allow some occasional adaptive mutations while maintaining low levels of polymorphism in this gene. The analysis on signatures of selection based on the Pfrh5 Pfrh6, all the Pfrh5 values were negative in all the study sites, and having negative values for the Pfrh5, this reveals that the gene is likely undergoing a prefined selection. But also, we also assessed the statistical tests that to test if the Pfrh5 results obtained were unreal, and all the Fnric and Fnric-F were also negative in all sites, indicating that the parasite is undergoing refined selection under this gene. Also, the linkage disequilibrium were also low, were as low as 0.06 in Heza, and as high as 0.32 in Morogoro urban. So having basis of negative touch mass values and low levels of linkage disequilibrium, this indicates that mutations within the Pfrh5 gene are rapidly eliminated, meaning that the parasite under this gene is allowing some occasional mutations, and it is also eliminating the non-advantageous mutations. That's why we see the domination of rare variants and the pattern that is consistent with preference selection or probably population expansion. So to conclude, based on our findings, having seen that there were a lack of population structure, having negative touch mass values across all sites, having low levels of nucleotide and haplotype diversity, we finally conclude that our findings suggest that the Pfrh5 gene is highly conserved across the sampled Plasmodium falciparum populations, despite that they were drawn from different geographic settings. But also its conserved nature likely is due to its crucial role in parasite survival, as we have seen that the gene plays a key role in inversion of the merozoites into the human erythrocytes. So from its crucial role in parasite survival, this likely facilitates its conserved nature within parasite population. So we just did not rely on our findings. We also recommend other findings. More important that other findings, including a broader geographical regions, should be conducted to validate the conservation of Pfrh5 and even assess its genetic stability, rather than relying on that at only 5 study sites. So some other studies, including multiple sites, should be done so as to assess the genetic stability of this gene. But also we recommend further studies using additional genetic metrics that were not assessed or were not included for our study. So the other genetic metrics can help to support the inclusion of the Pfrh5 into a broad malaria vaccine development platform. So I would like to give thanks firstly to the National Institute for Medical Research and my colleague from the Genomic Laboratory at the National Institute for Medical Research for their invaluable support, who were the co-authors of our paper. I really thank them for a high support in aiming to publish this paper and making everything being achievable until when the paper was published under Malaria Journal. But also thanks to the Pfrh5 Institute, who are the funders of the second phase of the molecular surveillance of malaria in Tanzania. Also thanks to the National Malaria Control Program in Tanzania. I also thank the malaria gene study collaborators who were able to collect data and submit data to the Malaria Genetic Epidemiology Network. Also thank to the Malaria Genetic Epidemiology Network for firstly inviting me to present the findings on this journal club, but also for putting the sample sequences accessible publicly so that are able to be accessed by each one and be advised to assess different genetic metrics. For mostly, I would like to thank the Bill and Melinda Gates Foundation, who are the co-founders of the project on molecular surveillance of malaria in Tanzania. Not to forget Prof. Deusi Shengoma, who is the project investigator under the project of molecular surveillance of malaria in Tanzania. Its support is really appreciated. I also thank everyone for his or her contribution. and everyone who is in this journal club. That marks the end of my presentation. Thank you all, and I welcome further discussions. Back for you, Pablo.
Thank you so much, Angelina. Great presentation, really well done on the pace, the way you explain, truly fantastic. And it's lovely to see familiar names on your slides of acknowledgement, people that we work with, so it's putting a smile on my face. Really great presentation. We've now reached time for discussion as well as Q&A. So please do raise your hands and we will allow you to speak, or type in the Q&A and chat box and I'll read them out for Angelina. While people are still thinking, I jotted down, I think, about 3 questions, and already one of your slides answered them, because I think your study is based in Tanzania. And I think one of your recommendations was that you are advising people to do further studies in different geographical locations. And I think that answered my question that I had about, will you do studies in different geographical locations? And I think that's part of your recommendations. The 2 questions that I have, the first one is, so Angelina, if a vaccine is developed, if a Pfrh5 vaccine is developed using your findings that you've provided in this presentation, how much more efficient do you think it's going to be compared to the R21 and RTSS vaccines? Because I think in your background of your study, you actually say that due to the limited efficacy of the R21 and RTSS vaccines, you do think that alternative vaccines need to be produced. So I think my question is, how much more efficient do you think your vaccine would be compared to those 2 that are currently out and approved by the World Health Organization? And the second question is, the R21 and the RTSS vaccines are mainly administered to children under 5, mostly in Africa as well. Who is going to be the sort of people that are given your vaccine if it's ever developed in the next 5 or 10 years? Do you have a particular age group that you are going to be targeting? Those are my 2 questions, Angelina.
Thank you, Pablo, for great questions. To begin with your first questions on the efficacy of the RH5 based on your findings. I really can't tell exactly how much the RH5 can offer protection, but based on the previous studies, their results really show that the efficacy of this vaccine could even probably be high than the efficacy of both R21 and the RTSS malaria vaccines. So indeed, I can't really tell that sure that the RH5 based malaria vaccine is really promising, given that it's highly conserved across multiple Plasmodium falciparum strains. Yes, for your second question, can you please repeat?
Oh, sorry, I should have asked them one by one. The second question is, so the R21 and RTSS vaccines are mainly administered to children. So if a vaccine is developed using the protein homolog 5, which is what your paper is about, who will it be targeted to? Who will be the main group that you think the vaccine should be given to?
I think, given that stemming from its crucial role in site survival, and another study was conducted in Tanzania, they evaluated the RH5-based malaria vaccine across different age groups, and they showed that the differences in efficacy was not that much, as it was comparable across children, across school children, and even across adults. So given that the efficacy was comparable across different age groups, so it might be targeted to different age groups with some variations in booster doses. So when the booster doses are given at different ratios, different age groups, it's going to offer a very high protective efficacy compared to the R21 and TSS, which are highly targeting only children and leaving other age groups.
Okay, thank you so much, Angelina. There are two questions coming in. The first one is from Dr. Richard Pearson. And Dr. Pearson says, thank you for the interesting presentation, Angelina. Did you run your own variant calling pipeline? If so, what was the pipeline, and did you compare the result of this with the variants found in the malaria gen Pfrh5 data release VCF files? That's a question from Dr. Pearson. Angelina?
The variants were called using the BCF tools, but unfortunately, we really did not compare the variant that we obtained from those that we obtained from the malaria gene datasets. I think we can put that under consideration for the next studies.
Thanks, Angelina. The next question is from Frank Jackson, who says, how did you account for mixed genotype infections when analyzing the Pfrh5 gene? Angelina?
For the mixed infections that were obtained, especially in the sites of, let me go back to the slides. Here. For the mixed infections that were obtained, especially in regions in Kigoma, Morogoro, and Nachingwea in Lindi region, firstly, we excluded these peripheral infections from analyzing the haplotypes, so that we are sure that among the monoclonal sample sequences, how is the haplotype distribution based on the monoclonal sequences? Not sure if I really got the question right.
Okay, thanks, Angelina. We still have about 10 minutes to go. So as I say, you are allowed to raise your hand and we'll allow you to speak, or you can type your questions in the Q&A as well as the chat box, and I'll read them out for Angelina. So please do let me know if you have any questions or comments or suggestions. I see there's a question coming in. Rather a comment, I think. So Dr. Richard Pearson is saying, thank you for the previous answer, Angelina. Have you looked at the RH5 genetic variation in PF Upload Atlas? And there's a link there. If so, do your results look consistent with this? So there's in the Q&A, Angelina, I don't know if you're able to see in the Q&A, there's a link that Professor Pearson has shared, and is asking if your results look consistent with this. It's the PF Haplo Atlas app, which came out sometime last year. Are you able to open the link and advise, or do you know about?
I've just heard about the atlas, but I didn't compare our findings from this atlas. Maybe I'll try to assess the diversity based on this atlas and then see if there are variations. Thank you so much for the link. We'll try to see if there are some variations based on our findings and the findings from this atlas.
Okay, thank you so much, Angelina, and thanks, Dr. Pearson. We have about 9 minutes left still, so questions, comments, suggestions, links to resources, you can continue to share in the chat or the Q&A. Please do raise your hand, and I'll allow you to unmute and speak. Going once. So I saw some names, and I see that Professor Ishengoma is in the room. Professor, do you have any comments on your side? I'm putting you on the spot here. Is there anything else that you'd like to add to the conversation? We have about 8 minutes to go. Okay, let me see. I see there's a question, I think, in the okay, Professor Dias, go ahead.
To give some explanations or to give my comments in the Q&A about the efficacy of different vaccine candidates. You asked whether the vaccine based on this gene could be more efficacious or less than the R21 or RTSS vaccine. But I just wanted to remind you and the listeners that the efficacy of a vaccine does not only depend on the nature of the gene, whether it's conserved or not, but it also depends on other things like the adjuvant, the ability to elicit antigenicity. So people with experience in vaccinology should be able to tell us that you cannot rely on the fact that the gene is highly conserved to predict that it will have very high efficacy compared to others, which are less efficacious or less conserved or highly variable, like CSP, which has been shown based on Beatus's results that you have high variability, a large number of haplotypes, and you get actually high efficacy when you have 3D7 strain and lower when you have other strains. So we cannot predict, but what these results tell us is that at least the gene is highly conserved. So if it does perform well in eliciting antigen response to the body, and again, if we are able to strike a deal to get the best adjuvant, which can be used with it, and probably it can work well. So it depends on the performance once the adjuvant is made and administered to patients. And again, whether you can use it in different age groups, what happens is you have to test, you have to do the trial and test in different age groups. I personally believe RTSS and R21 can do very well in other age groups apart from children, is simply because they haven't been tested. So, but we have evidence to show that they can do very well. So the question of whether the drug or a vaccine or a product can work in different age groups depends on the research evidence. It doesn't depend on the nature of the product alone, but on the evidence you generate once the study is done. I think I saw the paper, a recent paper on CSP and also with predictions. And I think there have been more studies, and I think malaria gene database is making a strong impact. I think in the next couple of, looking forward, and Richard is here, we should be able to map multiple genes and show whether, which genes, and based on predictions, which genes on their own or in combination could probably give us a better vaccine. So I think one of the reasons the genome studies and genome results have not been fully utilized is that we have not made strong, high impactful analysis to show which genes are more likely to give us better results. And this is probably underutilization of incredible results we have in initiatives such as malaria. So let me end here to give us others a chance, but I think we now have an opportunity, given the analytics capacity, to do more and much better than we have done in the past. Over to you.
Thank you so much, Professor Dias, for your contribution. I think, yeah, you did add more to the questions that I had earlier. Thank you so much for explaining further to what Angelina did earlier. Thank you so much, Prof. I see Frank Jackson has another question in the chat. Frank is asking Angelina, were there any polymorphisms identified that are known or suspected to reduce the efficacy of Pfrh5 based on invasion inhibition? Angelina?
For a start, we did not assess the polymorphisms at specific codons around the gene. Instead, we just analyzed to see the overall levels of polymorphism within the gene. We did not assess at specific codons, so we can't really tell if they were comparable to other previous studies.
Thanks, Angelina. We have about 2 minutes to go. Any burning questions, any burning comments before I release all of you? Going once, going twice. Okay, any last comments from yourself, Angelina?
Maybe to thank the audience and to thank all who have raised some questions and even all who have contributed to this research. And so just that, I thank everyone. I thank the malaria Gen for inviting me to share and highlight some of our key findings from our published paper. So the paper is already published and it is on malaria journal. So everyone can have access on it and read and try to understand. And if one can have questions, I'm open to communicate and even to continue the dialogue out from here. Thank you.
Thank you so much, Angelina. We can share your email with people and they can connect with you on LinkedIn. I saw you have a LinkedIn account as well, so they can connect for collaboration and if they have further questions. Really great presentation, Angelina. Good luck with your MSc at the University of Basel. I think you have a great future ahead of you as a scientist in the malaria space, and you are making great contributions to helping eradicate malaria in our lifetime. To the audience, thank you so much for joining us for the April Journal Club. We are going to have another one at the end of May. Be on the lookout on our social media as well as our newsletter. Please sign up for the Malaria Gen newsletter and you'll hear about what the topic is and who the speaker is as time goes. We will send out invitations and links for registration for that as well. But yeah, thank you so much for joining us. Have a fabulous day, everyone, and see you next month. Thank you so much, everyone.
Thank you.
Thank you too, and see you soon.